Have you ever wondered how we can reduce our dependence on fossil fuels? What if I told you that the answer lies in something that's small enough to fit in the palm of your hand? Hi everyone, welcome back to my channel. My name is Julian Wiggins, and in this video we're going to talk about a fascinating topic, which is how we can use bacteria as a source of biofuel. But you might be thinking, wait, what? Bacteria can produce fuel? Yes, you heard that right. In fact, scientists have been exploring this idea for quite some time, and the results are extremely promising. So let's just dive right in and learn more about it. Biofuels are fuels produced from renewable biological resources, such as crops, animal waste, or algae. They're often used as a substitute for fossil fuels in transportation, heating, and power generation. The advantage of biofuels over fossil fuels include renewability, which means biofuels are produced from renewable resources, as I mentioned before, which means that they're sustainable and can be replenished over time. They also reduce the greenhouse gas emissions, so when they're burned, biofuels produce lower levels of greenhouse gases than fossil fuels, which helps reduce climate change's impact. Improve air quality, biofuels burn more cleanly than fossil fuels, producing fewer harmful emissions that can lead to respiratory problems and other health issues. Energy security, by using domestic sources of biofuels, countries can reduce their dependence on imported oil and improve their energy security. And finally, job creation. The production and distribution of biofuels can create jobs in rural areas, which can help to stimulate local economies. Overall, biofuels are a promising alternative to fossil fuels because they offer a renewable, sustainable, and environmentally friendly source of energy. Bacteria can produce biofuels through a process called bacterial fermentation. During fermentation, microorganisms such as bacteria break down organic compounds, such as sugars or starches, into simpler molecules, like ethanol, butanol, or hydrogen. But let me go into a more detailed explanation of how bacteria can produce biofuels. First is feedstock preparation. The first step in bacterial fermentation is the preparation of the feedstock. This can be any organic material that contains sugar or starch, such as corn, sugarcane, or cellulose. Enzymatic hydrolysis, if the feedback is a complex carbohydrate like cellulose, it needs to be broken down into simple sugars. This can be done by treating the feedstock with enzymes that can break down the complex carbohydrates into simple sugars such as glucose. Next is fermentation. After the feedstock has been prepared, it is then mixed with a bacterial culture and a mixture is allowed to ferment. During fermentation, bacteria metabolize simple sugars and convert them into biofuels, such as ethanol, butanol, or hydrogen. For example, ethanol production from bacterial fermentation involves the following steps, all taken from the article by Kessler et al. First is the conversion of glucose to pyruvate, which is when the bacteria convert glucose to pyruvate through a process called glycolysis. Next is conversion of pyruvate to ethanol, which the pyruvate is then converted to ethanol through a process called alcoholic fermentation. And finally, what is performed is separation and purification, which happens after fermentation, which is the biofuel product, in this case ethanol, is separated and purified from the fermentation. Similarly, butanol production involves the conversion of butyric acid to butanol through a process called acetone butanol ethanol, or rather ABE fermentation. The ABE fermentation process involves the conversion of carbohydrate to butyric acid, and then to butanol through a process of chemical reaction. Finally, hydrogen production involves the conversion of organic compounds to hydrogen gas through a process called dark fermentation. During dark fermentation, bacteria break down organic compounds into hydrogen gas and other byproducts. In summary, bacterial fermentation is a process that can be used to produce biofuels such as ethanol, butanol, and hydrogen from organic materials containing sugar or starch. The process involves the conversion of simple sugars to biofuels by bacterial production. There are several types of bacteria commonly used for biofuel production, each with its unique advantages and limitations. Here are some of the most commonly used bacteria for biofuel production. The first is Escherichia E. coli. E. coli is a well-known bacterium used in various biotechnology applications, including biofuel production. E. coli is commonly used to produce ethanol through a process called homolactic fermentation, where glucose is converted to lactic acid and then to ethanol. However, E. coli has limitations in terms of substrate utilization and low ethanol yield. The next is Clostridium. Clostridium is a genus of bacteria that can produce a variety of biofuels, including butanol and hydrogen. Clostridium is commonly used for butanol production through a process called ABE fermentation, where acetone, butanol, and ethanol are produced from carbohydrates. Clostridium can also produce hydrogen gas through a process called dark fermentation. However, Clostridium has limitations in terms of low productivity and sensitivity to environmental conditions. Cyanobacteria, also known as blue-green algae, can produce biofuels through photosynthesis. Cyanobacteria uses sunlight to convert carbon dioxide and water into organic compounds and oxygen. Cyanobacteria can produce a variety of biofuels, including ethanol, hydrogen, and biodiesel. However, cyanobacteria have limitations in terms of low productivity and susceptibility to contamination. Other bacteria used for biofuel production include Xyomonas mobilis, which was used by Ingram Lowell et al., and which is hers, it was commonly used to produce ethanol, and Rhobacter spiraeus, which is used for hydrogen production. This was achieved by using the genes that encode for the pathway, and by using numerous methods in the procedure. For the genetics methods, they used plasmid DNA that was isolated and purified through a separate laboratory. Overall, the choice of bacteria for biofuel production depends on various factors such as the type of biofuel to be produced, substrate used, and the required productivity and yield. While there is still much research to be done to optimize the use of bacteria in biofuel production, these microorganisms show great potential for sustainable energy production. Scaling up biofuel production using bacteria presents several challenges that need to be addressed to achieve commercial viability. Here are some of the challenges faced in scaling up biofuel production using bacteria. The first is feedstock availability and cost. The cost and availability of feedstocks such as corn, sugarcane, and cellulose are critical factors in biofuel production. Scaling up biofuel production requires large quantities of feedstock, which can increase competition with food production and results in high prices. Finding alternative and sustainable feedstocks such as agricultural waste could help address this challenge though. The next is fermentation efficiency and yield. The efficiency and yield of bacterial fermentation determine the amount of biofuel produced. Optimizing bacterial strains and fermentation conditions can improve efficiency and yield, but this requires extensive research and development. Next is contamination and process stability. Bacterial fermentation is vulnerable to contamination by other microorganisms, which can reduce yield and quality. Ensuring process stability and preventing contaminations requires careful monitoring and management of fermentation conditions. Next is infrastructure and logistics. Scaling up biofuel production requires significant infrastructure and logistics, such as facilities for rest stock storage, fermentation, and periodic separation and purification, which can be a significant barrier to commercial viability. And lastly is just the understanding of the network of genes, proteins, and metabolites that you're never able to do this. In a paper by Mokohade, Andrila et al. About the importance of systems biology and engineering microbes for biofuel production, these challenges include the complexity of microbial metabolism, the need for large experimental data, and the difficulty of predicting the behavior of engineered strains in real-world environments. But despite these challenges, there are several potential opportunities for scaling up biofuel using bacteria. First is sustainable energy production. Biofuels produced using bacteria have the potential to replace fossil fuels, reduce greenhouse gas emissions, and mitigate climate change. Next is rural development. Biofuel production can provide economic opportunities for rural communities through feedstock production and processing. Then there's circular economy, where biofuel production can be integrated into a circular economy where waste products from other industries such as agriculture and forestry are used for feedstock for biofuel production. And lastly, there's biorefineries, where biofuel production can be integrated with other biorefining processes, such as the production of chemicals and materials from biomass to create a more efficient and sustainable use of resources. In conclusion, scaling up biofuel production using bacteria presents several challenges, but also significant opportunities for sustainable energy production and economic development. Continued research is, however, still needed, and development are essential to overcome these challenges and fully realize the potential of this technology. And there you have it, folks. Bacteria might be small, but they have the potential to play a big role in our energy future. With their ability to produce biofuels, we can reduce our dependence on fossil fuels, decrease greenhouse gas emissions, and promote a more sustainable world. So let's keep supporting scientific research in this field, and who knows, maybe one day we'll be fueling our cars and homes with bacteria. But as always, thank you for watching. My name is Julian Wiggins, and I hope to see you in the next video.